Biology 121 Online. This is our 16th and final video. Today we'll talk about genetic variation within a population. We're going to be talking primarily about human populations within this video because that's what most of us think most about. But a lot of these principles do apply to other species of plants, animals, and fungi especially. So let's talk about some key vocabulary. First, we have to identify a population. When we talk about a population, we're talking about a group of organisms that are of the same species. There's a concept in ecology called a community, which assembles populations from different species, but we're going to focus just on populations today because we're talking about genetic variation, so let's stay within a single species for the most part. A genome is the complete DNA sequence of any one individual. So you've got a genome and I've got a genome and your sister has a different genome than you unless you're an identical twin, and your mother has a different genome than you unless you're a clone. So everybody's got a genome that refers to the complete DNA sequence of a single individual. A gene refers to the sequence of DNA that forms a particular protein recipe. If you think back to our transcription and translation lessons, where we talked about how DNA is expressed. When DNA is expressed, what that expression is the formation of a protein according to the instruction set that's built into the DNA. So, a gene is a sequence of DNA that forms a particular protein recipe. A gene loci is actually plural. A gene locus, or plural, gene loci, refers to the specific places, the locations, if you will, within a DNA sequence at which copies of the same gene are found. So there's lots of genes that are really important. And if a gene is really important, your DNA genome probably contains multiple copies of that recipe, and they may actually be different copies in different places within your DNA. They may vary from each other. What we call it when one variant of a gene is different from another variant of the same gene, that's called an allele. So alleles are specific variants of the same kind of gene. So if you have a gene for producing, let's say, amylase, the digestive enzyme that's in your saliva, and I have a different version of the amylase gene, we would say that my allele is different than your allele. An allele dosage refers to the number of copies of the same allele that you have in your genome. So that means, like, if you've got 6 different loci for the same gene, and 3 of them carry one allele, and 2 of them carry a different allele, and a third one carries yet a third allele, then we'd be talking about those different copies of the same alleles scattered throughout your genome as the allele dosage for each of those variants. A nucleotide is a single piece of DNA. That's our G's, A's, C's, and T's, guanines, adenines, cytosines, and thymines that form up the individual units of DNA. And so if that's what a nucleotide is, a single nucleotide polymorphism is where you have the smallest possible difference between two alleles, where you have a change. A polymorphism means a different or many forms. So if you have many forms of a single nucleotide, that means that in one person's allele you have a variation in a single nucleotide within that allele that makes this allele different from another allele. So we're going to talk in a moment about alleles as dominant and recessive as well, but we'll get to that. So first, a couple of fun facts, specifically looking at humans. A human's genome contains about 3.2 trillion nucleotides. This is thousands, millions, trillions. 3.2 trillion nucleotides. These form up 25,000 genes, we think. There might be some we haven't found yet, and there's some debate as to about 4,000 of them, but it's somewhere between 20 and 25,000 genes that we know about right now. Each of those genes varies in length. They can be as short as about 300 nucleotides, and they can be as long as 2 million nucleotides. Obviously, what that depends on is the size of the protein that they code for. And we should note that only 2% of the nucleotides in your DNA, in your genome, are part of genes. The rest of them are in non-coding sequences. Now, if you go back and remember when we were talking about DNA transcription, we said that within the DNA sequence at a particular gene, there's going to be some parts of the DNA that don't actually participate in the recipe, and you got to cut those ones out. We called those introns because they're inserted inside between the coding parts of the DNA, and they have a variety of functions. There's reasons why there are introns there that have to do with expression and regulation, and also with the fact that some genes may actually carry information that is used in different recipes for different proteins. And so some of those non-coding sequences are introns, some of them are structural pieces that ensure that when the DNA is wrapped around a histone, the expressive part of the DNA is on the outside and the non-coding sequences are on the inside. So there's a lot of structure to the DNA that accounts for a good part of that 98% of what they used to call junk DNA, but now they more accurately call simply non-coding sequences. Some genes are repeated at hundreds of different loci. So in some cases, you might have a gene that's so important, your body maintains hundreds and hundreds of copies of it. When I was looking up some information to try to get a sense of what's the highest known count of what we call polygenics. So when a gene is repeated at more than one loci, we call it polygenic. If it only exists one time within the genome, we call it a monogenic trait. But some of these, one of the studies I found on polygenic traits was looking at 180 different loci that all contribute to making sure that your skeleton gets built. Right? That's an important part of being a human being that is alive. You gotta have a skeleton. So there's hundreds of loci that participate in making sure that that work gets done correctly. In some cases, when genes are located near each other, they may work together. So when one gene gets expressed, it can stimulate the expression of another gene, or one gene that is located close to or even overlapping with. Remember the non-coding sequence and introns, right? So when two genes are overlapping with each other, that can have an effect on one of them getting expressed relative to the other, and so there's associations between genes that are similarly located within the DNA. And this is the fun part, all humans share 99.9% identical DNA. Now that still leaves, I've got this number wrong. Yeah, okay, so there's supposed to be another 3 zeros here, but it wouldn't have fit anyway. So this should have been 3.2 trillion. I think I've made it thousands, millions. Yeah, okay, so there should be another 3 zeros up here, and there should be another 3 zeros down there. So there's still 3.2 billion single nucleotide polymorphisms that can vary within that 99.9% identical DNA. Now, remember also that every single nucleotide polymorphism could be any of the 4 different nucleotide bases, G, A, C, or T. So if you think about like 4 to the power of 3.2 billion, or even, yeah, I think that's the way it would go. It might be 3.2 billion to the power of 4. That's still a number so large that while it is true that it is like, there is a finite number of different human DNA sequences that could exist, but that number is so large, it would take me a long time to write out the number of zeros at the end of that one. So for comparison, by the way, if humans are 99.9% identical to all other humans, humans are 98.8% identical to chimps, right? So chimpanzees are a related species of primate with a relatively recent common ancestor to humans, probably about 3 million years ago. And chimps, bonobos, and gorillas all share quite a large percentage of their DNA with humans, right? So we actually have a relatively strong amount of our DNA identical with chimps. But the reason we can easily distinguish humans from chimps is because if we think about this, we are 0.1% different from each other, and we are 1.2% different from chimps. So that's, 120 times more variability? It's a lot, right? Okay, let's talk again about gene loci. A locus, which is just Latin for location, describes the specific location of a gene on a chromosome. So if we look at the picture at the bottom here, we have two homologous chromosomes, right? That's what the little arrow here is helping us to recognize, is that, oh, it's a chromosome. And we know they're homologous chromosomes because it says right there. And also because they happen to be exactly the same length and have their narrowing spot at exactly the same place. This is called the centrosome, by the way. And then there's these gene loci. And what we can see is there's two copies of this P gene on each chromosome. There's two copies of an A gene on each chromosome. And there's one copy of a B gene on each chromosome. And so these alternative versions of a gene reside at the same locus. You'll see that there's a location on this chromosome and its homologous chromosome where a P gene is located. There's a location here on both chromosomes. It's in the same spot on the chromosome. And that's why we call them loci, locus, location, right? So the two chromosomes can bear identical alleles at each locus, right? Here we have a big P and a big P, so those represent two different alleles. The dominant allele for each of those cases that this individual received from, let's say, the mother. But from the father, this individual received one dominant allele and one recessive allele at the P loci. So same thing with the A's. They could be the same or they can be different. And the B's from one homologous chromosome inherited from one parent to the other homologous chromosome inherited from the other parent. Obviously, there's no reason why these would have to match. It depends on what the genotype of the parents were. So if we take this all together, our total genotype is AA, right? So one capital A and three lowercase. This is a capital A with a lowercase a, capital A with a lowercase a capital B with a lowercase b, so one dominant allele and one recessive allele for the B gene, and then two dominant P's and a dominant P with a recessive P. And in the standard way of doing this, we associate these together according to their locations. So we're going to put this capital A with this lowercase a, that's my first AA, and then we got in the other location two lowercase a's. So if we talk about the allele dosage of the dominant gene, on the A gene, we have one dominant allele. On the B gene, we have one dominant allele. And on the P gene, we had three dominant alleles. So in some cases, the number of dominant alleles or the number of a particular variant of allele that you have will affect the genetic expression. This happens with things like height and eye color and skin tone and hair waviness. So there's lots of these traits where you see a wide variation between people, and that can be partially explained at least. Some part of the explanation for that is that you have these allele dosages dependent upon multiple different loci. So when we have two loci, this is where it gets real complicated real quick, and why we didn't really mess around with Punnett squares in this class like you did back in 9th grade. Because if we have our parent allele, they're going to have, at two loci, they're going to have two different sets of, right? So in this case, the A, the B, the C, and the D might be like a capital P, a capital P, a capital P, and a lowercase p. But we're just saying, I mean, look, there's four variants. That could be all four, right? And the other parent is going to contribute is going to carry, four different alleles, or possibly multiples, possibly some of these are identical alleles. And when we cross these like a Punnett square, we might have A and C with E and G, that's our first one here. Or we might have A and C with E and H, that's our second one. Or it might be A and C with F and G, there's our third one. And we can continue on through this until we've generated all 16 possible outcomes, some of which may be identical or functionally similar to each other, because I haven't identified, like the A here might actually be exactly the same allele as the C there. I was just breaking it out to show all of the different possibilities. So in general, the combinations are going to be four, because there are four different combinations of bases. to the power of L, the number of loci. So in your freshman year Punnett squares, there were always 4 squares inside there, right? Because you were only dealing with 1 gene locus. When we go to 2 gene loci, as we've just shown, we get 16 possible locations. If we get up to 7 gene loci, we have 16,384 possible different variations. And as I just said, some of these things are hundreds. So if you think about like what's 4 to the power of 100, it's a big, big number. So that accounts in part for the wide range of variation in human phenotypes. Now, let's talk a little bit about human migration patterns and the effect that has on variability of DNA within humans. So, Homo sapiens as a species evolved in Africa about 200,000 years ago. And then started spreading in to spaces that were previously occupied in West Africa and throughout the Middle East, occupied by Homo erectus. Homo erectus is an ancestral species that that went extinct sometime. I think Homo erectus went extinct before Homo sapiens even evolved. There is some intermediary species that we don't even have fossils of yet. So Homo erectus dwelled throughout Africa and the Middle East and into South Asia, and was after it went extinct, Homo sapiens began moving into West Africa and moving up into these parts of southern of the Arabian Peninsula and into Europe, where they encountered a group of Homo neanderthalensis that had already left Africa about 200,000 years ago, around the same time that modern humans were evolving. And so these modern humans, Homo sapiens, interbred with Homo neanderthalensis, forming the European modern humans. They also moved further out into Asia. There's some evidence that some stragglers of the Homo erectus species were still living in sort of Southeast Asia, and may have interbred with modern humans as well, forming a new species, the Homo floresiensis. But eventually, 70,000 years ago, we see this migration pattern into Asia, and then up into what are now the cold climes of Russia, but at the time were not so cold, 25,000 years ago, down into the Polynesians and Australia about 50,000 years ago, and up into the Bering Strait and across the land bridge that formed between Siberia and Alaska 15,000 years ago, as a group of Asians known as Asian modern humans, known as the Beringians, came through Beringia, now the Bering Strait, and populated North America. So as a group of humans migrates from one area to a new area, they only carry certain alleles with them. This is called the founder effect. So if we think about the parent population here having looks like 1, 2, 3, 4, 5 different kinds of alleles, right? And so each of these dots represents an individual and a particular different gene variant or allele that individual is carrying. But only some of those individuals from that population move into a new space. And then if they become isolated from the parent population where they came from, then you're going to see less genetic diversity in that new population. So if we think of this as like the Asian Beringian population with these many different alleles, only some of them crossed the Bering Strait, after which the ocean levels rose again and cut them off from their ancestors, from the other descendants of the same ancestors still living in Asia. And so you have a smaller amount of genetic diversity in humans that were native to North America after 15,000 years ago, but in pre-Columbian times. But those variants are mirroring a small subpopulation of the Siberian and North Asian population. So the variation in which alleles are present within a population can tell us something about that population's migratory history. DNA companies, like the ones that'll like swab your cheek or spit into this cup and send it back in, and they'll do an ancestry DNA test on you. They claim to tell you that this effect can also tell you things about an individual's ancestry, but that's a lot more complicated, especially as in the last 2,000 years, humans have migrated much, much more rapidly. And so the people who live in a place now may not be the same people who were living in that place even 100 years ago. So if you get a DNA ancestry test that says, oh, you're related to people who now live in this place, that doesn't necessarily mean that that's where your ancestors came from. Finally, when we trace family relations, since we've just talked about ancestry DNA tests, there are some things that we can do that are reliable and stable ways of telling who's related to who. So, sexually reproductive organisms receive their mitochondria only from their mothers. Remember, with a sperm cell and an egg cell, the egg cell is much, much larger. And so it can carry mitochondria, whereas the sperm cell doesn't really have room for that. So mitochondria have their own DNA, and unless there's a mutation, mitochondrial DNA is transmitted unchanged matrilineally. That is to say, you will have the same mitochondrial DNA, whether you're a boy or a girl, as your mother, your grandmother, your great-grandmother, and all the way back until the point at which some mutation occurred in the mitochondria within an egg cell. Again, it has to be a germline mutation in order for it to be inherited. For that reason, there's really only 8 major groups of mitochondrial DNA in humans. There's smaller differences between mitochondrial DNA that can also tell us more about specific family lines matrilinearly. Then we say that we note that sexually reproductive XY organisms, like humans, have an X chromosome and a Y chromosome. There are other kinds of sexually reproductive chromosome sets, but for XY organisms, we only receive a Y chromosome from the father. So unless, again, there's a germline mutation, Y chromosomal DNA is transmitted unchanged patrilineally. That is to say, every male has a Y chromosome that should be identical to the father, the grandfather, the great-grandfather, and so on back. Everything else gets mixed by recombination during meiosis. And I should say, Y chromosomal transmission does get a little bit dicey because the Y chromosome and the X chromosome in a male also do get remixed by recombination during meiosis, but because of the shortened length of the Y chromosome, that crossing over that causes the recombination effect doesn't happen as much in the X and Y chromosomes as it does in the other chromosomes. So it's a lot more stable, and it's not as stable as mitochondrial DNA, but it does give us something to work on in terms of patrilineal relationships. And that's why if you're someone who watches those shows where they do a paternity test, they say with 99.9% probability, you are the father, right? Because there's a small chance that the Y chromosome DNA got intermixed with X chromosomal DNA, but it's pretty likely. So that concludes our final video in the series. Please do let me know if you have questions or concerns, and I'm looking forward to the wrap-up of the wild conclusion of our class this year.